All right, so I want to continue talking about the nervous system. We're going to be moving on today to talk a little bit more about neuron physiology and the action potential. This is the first part of it. And so we talked a lot about what a neuron was, what nerves are, what the brain and the spinal cord are. And now we're going to move on to some of the more physiology of how exactly, the nervous system does some of its things like sensing and transmitting information. And so the main things that neurons do is that they receive stimuli either from outside the body, right, or from another neuron, and then they transmit electrical signals throughout the body. And they translate, so like let's say you're having a sensation, like a feeling or a smell or a sight. They translate all of these sensations around you into electrical signals, and that they can then send up to the brain. And then this is important because your brain can really only interpret and it deals with and it sends out electrical signals. And so that way, everything is sort of translated into this neuron electrical signal language. And so your book goes into a little bit about Ohm's law and the neurons, and they're kind of talking a little bit about electricity and electrical current and how that relates to neurons. And I think that is definitely something that you should read if you have a little bit of background in understanding the physics of electricity and electrical current. But if you don't, I think it might be a little bit confusing. So for the purposes of this class, I'm just going to mention that an electrical current is a stream of charged particles, such as electrons or ions, and we're going to be talking about ions in this case, moving through an electrical conductor or space. So much like electrical current flowing through a copper wire in your house, the movement of charged particles in the neuron creates a current in the neuron. And we call this electrical signal an action potential. And so we're going to be talking about the action potential and how it's made and how it's propagated down the neuron and how the neuron is then able to, transmit that electrical signal to another neuron and so on. And so in order for the neuron to transmit its signal from one to the next, right, there's they have to connect somewhere. So synapses are the places where neurons sort of connect or come really close to another neuron or an effector, right? Because let's say we have a motor neuron going from the brain down to our skeletal muscle, the neuron is going to have to talk to the skeletal muscle. And if you recall, we talked about this already in our neuromuscular junction, right? So the neuromuscular junction is a great example of a synapse, right? So it's a synapse where one nerve is meeting a muscle and the nerve is sending its information, its electrical signal, to the muscle. And so there are two types of synapses. The first one is a chemical, and chemical synapses are by far more common than electrical synapses. And we have already learned about chemical synapses, even if you didn't realize it, because the neuromuscular junction is a great example of a chemical synapse. And then the other type is an electrical synapse. The electrical synapse, we're not going to spend as much time on because it's really a lot less common. But the electrical synapse is basically when the actual neurons are connected to each other. So we have the two neurons and they're actually touching and they're actually connected to each other, and they have gap junctions in between them. And remember, a gap junction is where we have our cells, and our cells are actually connected, and the gap junctions have a little protein pore so that they can share cytoplasm, and ions can move directly from one cell to the next. And so these gap junctions and these electrical synapses occur between some neurons, and then cardiac and single unit smooth muscle also are connected by gap junctions, and so we can liken that to an electrical synapse. And we'll talk more about cardiac muscle and single unit smooth muscle soon, right after we get to the autonomic nervous system. So an advantage of an electrical synapse is that it is really fast, right? We have the ions moving directly from one cell to the next, and there's no need for a neurotransmitter. There's no delay in passing the electrical signal. But a con is that they can't integrate information and make decisions. So if we have an electrical signal coming from one neuron and it's attached to the next neuron by a gap junction, it has to go. There's no really regulating it. Whereas we'll talk about with chemical synapses, we can have multiple different inputs converging on a single neuron, and then those, depending on like how many excitatory inhibitory inputs it gets, the next neuron can sort of, decide if it's gonna, if the action potential is going to happen. And so the chemical synapse is more useful for that. So what is a chemical synapse? We have a couple of vocabulary terms that we need to talk about so that we're all on the same page. A synapse is going to be the area in between the two neurons. And the presynaptic neuron, right? Presynaptic is the neuron that is initially sending the information. Usually it's drawn at like the top of the page or the left side, right? That's where the information is coming from. That's the neuron that produces the signal. Then we have the synaptic cleft, which is a small fluid-filled gap between the two neurons, because in a chemical synapse, they don't actually touch. They're actually like very, 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 very close, but they're not actually touching, so they're very close. And that synaptic cleft or synaptic gap is going to be that small fluid-filled space between the two neurons. And then on the other side, the neuron that's receiving the signal is called the postsynaptic neuron. And either the cell body or most commonly the dendrites are going to be the part of the new neuron that's receiving the signal. And so if you think about what happened at the neuromuscular junction, that's going to be the events that normally happen at a chemical synapse. So neurotransmitter molecules are released from vesicles in the synaptic knob into the synaptic cleft. The neurotransmitter diffuses across the cleft and binds to postsynaptic receptors, right? In the motor end plate, it was the acetylcholine receptors, but we have some other types of receptors that we'll talk about that we could possibly have in neurons. The binding of the neurotransmitter to the receptor is going to initiate what we call a postsynaptic potential or a graded potential, and we'll kind of unpack those terms in a little bit. And this may trigger an action potential in the postsynaptic neuron. And the synaptic delay is the time it takes for all these things to happen. Like we said, chemical synapses are a little bit slower than electrical synapses because like that neurotransmitter and everything is involved. We have to have a bunch of stuff happening. Whereas with the electrical synapse, you know, they're connected and the ions just move from one cell to the next. So this is not from your book, but it's another picture of just a chemical synapse right here. We have the presynaptic neuron right here. This is the axon, the synaptic knob of the presynaptic neuron. It has these nice little synaptic vesicles, and they're releasing neurotransmitter into the cleft. All right, these guys are going to be the receptors on the other side of the postsynaptic neuron. Then these receptors will cause a change to happen in those receptors, which will eventually cause some sort of graded potential or some sort of effect in that postsynaptic neuron. So one thing I want to mention is, like, neurons are big, right? So the nucleus is up here, and all of like the Golgi and the rough ER and everything are up here. So how do the neurotransmitters get down to these synaptic vesicles? And you know, how do any waste products get back up? Because this is a really long cell, and so we want to talk a little bit about neuron transport. So the axons are really long, and so we have to have transport up and down the axons because, you know, most of the cellular machinery is going to be found up in the body or the soma. And so we call something anterograde transport if it moves from the cell body towards the synaptic knob. That's kind of like, I don't know, the right direction. Whereas a retrograde transport is when we have things like waste products and used materials from the synaptic knob going back to the cell body. And that's kind of like, I think it's moving like backwards. So retrograde transport goes back to the cell body. And so these two types of transports can either happen fast or slow. And so there are two different types of movement. So fast axonal transport occurs at about 400 millimeters per day. And I think of it as like train tracks. So it involves movement along microtubules. Remember, those are going to be the biggest type of cytoskeletal elements. And so we have movement along these, like, I think of them as train tracks, the microtubules, and they're powered by motor proteins that split ATP, right? So that's like the motor or engine on a train. And so it can move up or down the train tracks, right? And anterograde or retrograde motion is possible here. And so we're going to take vesicles and organelles and glycoproteins from the cell body down to the synaptic knob on this microtubule train track, right? That is powered by this ATP engine, like a train. And then we also, the train tracks we have another set, you know, that comes back and it goes in the other direction. And so the retrograde transports, we have another train that's going to go from the axon back up to the cell body. And it transports the used vesicles or any potentially harmful products or waste products and things back up to the cell body. And so that happens fast and it's bidirectional, and I think of it as like train tracks because it needs a motor and you need the microtubules like a train track. Whereas this next one is called slow axonal transport. And this one is a lot slower. It occurs only at 0.1 to 3 millimeters per day in contrast to the 400 millimeters per day. And it results from the slow flow of axoplasm, and that's generally going to go from the cell body to the synaptic knob, and so we only have anterograde transport this way. And I think of this as like a very slow-moving stream, and you're kind of floating on a raft down the slow-moving stream, or maybe you're just floating down the slow-moving stream. And so the substances are slowly moved, and they can only go in one direction, which is the direction of the current. So enzymes, cytoskeletal components, and new axoplasms will be moved that way. So this is just kind of interesting how the communication happens in the neuron, like how we move the neurotransmitters like from the body down to the synaptic knob, right? Because all of the stuff, like the Golgi apparatus and the rough ER and everything that makes a lot of these proteins that are needed for some of these neurotransmitters are going to be up in the body, and then they have to transport them all the way down to the synaptic knob, which can be pretty long distance sometimes. All right, so now we're going to move on to a little bit of neuron physiology. And so in order to understand how an action potential is created, we're first going to talk about a few things. The anatomy of the plasma membrane of the neuron, right? This is important. This is why we reviewed membrane transport. And then what happens in each part of the neuron. So the action potential is very similar to the first and second steps of muscle contraction, right? So the neuromuscular junction and excitation contraction coupling. So hopefully you'll find some of this material familiar, right? And this is going to hopefully also reinforce the concepts we learned in chapter 10. All right, so the first thing I want to review is the types of pumps and channels and the difference between a pump and a channel. I know it probably feels like we've reviewed this a lot, but it's important. All right, so pumps are membrane proteins that maintain a concentration gradient by moving substances against their concentration gradient. So from areas of low concentration to areas of high concentration. They require cellular energy to do this, just like pushing the bowling ball up the hill. Neurons have sodium potassium pumps and calcium pumps in their membranes. All right, the sodium potassium pumps are going to be everywhere, and the calcium pumps we'll see are going to be found in the synaptic knobs mainly. And then they also have channels. So channels are protein pores in the membrane that allow ions to move down their concentration gradients, right? So from an area of high concentration to an area of low concentration. This does not require energy, right? It's like that bowling ball can just roll down the hill by itself. And then when some channels are always open and some are closed, and they open momentarily and close again, but when they are open, they allow a specific type of ion to diffuse. So they don't allow everything to diffuse, only that specific type of ion. And so we're going to talk about sort of the entire plasma membrane of the neuron is going to have leak channels, like potassium leak channels and sodium leak channels, sodium potassium pumps. And these are going to be found everywhere, and they maintain the resting membrane potential. And remember, we have reviewed the resting membrane potential a few times. We're going to review it again now, just a little bit, because it is important. So remember that neurons at rest are going to have ions unevenly distributed across the plasma membrane. And these ions are going to be the ion that's inside the plasma membrane in the cytosol is mostly going to be potassium. So remember I said potassium, like think puppies. Puppies want to be inside the house. And so they have a higher concentration of potassium in the cytosol as opposed to outside the cell in the interstitial fluid. And they have higher concentrations of sodium chloride and calcium outside of the cell, right? It's called the interstitial fluid or the tissue fluid. And so make sure that you know where these ions are at rest. That will be a test question. And you need to know it just for the basis of understanding this. So there is also a calcium concentration gradient at the synaptic knob where we have more calcium outside the cell than inside. And so at rest, the gated channels are closed, and there is an electrical charge different across the membrane. This is called that resting membrane potential. So the cytosol or inside of the cell is relatively negative compared to the interstitial fluid. And the resting membrane potential of a neuron is typically around negative 70 millivolts. Whereas you think back to skeletal muscles, it was around negative 90, but either way, it's pretty close. This difference can be measured with microelectrodes and a voltmeter. All right, so if you recall, we talked about resting membrane potential in a previous video. You can go back and watch that again. I'm not going to go through the whole thing, but so we have sodium leak channels and potassium leak channels, and we have sodium potassium pumps. So our sodium-potassium pump is going to be really, really, really important in establishing and maintaining these large concentration gradients, right? We're putting 2 potassium into the cell. The puppies are going into the cell. We're putting 3 sodium outside of the cell. They're the snakes. We're throwing the snakes outside of the cell. Remember, we also, since we're only pulling in 2 positive charges, but we're getting rid of 3 positive charges, that's going to contribute a little bit to our negative charge on the inside of the cell. I liken it to a bank account. If you only make $2 a day, but you spend $3 every day, you're going to go negative pretty soon. We also have a bunch of negatively charged proteins inside the cell here. And so we will have some potassium leaking out and some sodium leaking in, but the sodium potassium pump is going to continue to pump those all the time, so we can maintain these high the high concentration gradients. This one does not talk too much about chloride and calcium, so recall that chloride is going to hang out near sodium, and so chloride is going to be outside also. And then calcium is generally always pumped outside of the cell, and so especially around the synaptic knob. So we're going to have more calcium outside around the synaptic knob. And so, as we said before, the role of sodium potassium pump, really important. It pushes 3 positive charges out. So 3 sodiums are going to go out. It only pulls 2 positive charges in. 2 potassium are coming in, and that's going to contribute a little bit to the negativity of the inside of the cell. But most importantly, it maintains the concentration gradients for those ions. And so here is another representation. We have sodium and chloride outside, and we have potassium inside. And then if we were near the synaptic knob, we would also have calcium outside. All right, so now we're going to talk a little bit about the different types or the different segments of the neuron. So there are 4 different functional segments of the neuron, and our functional segments of the neuron are going to be, we're going to go in order. The first one is the receptive segment, and so that's going to be the cell body and the soma, and excuse me, the cell body or the soma and the dendrites. And so these are going to contain special chemically gated channels. And so another term for chemically gated channel is a ligand gated channels. And these are going to be chemically gated chloride channels, chemically gated potassium channels, and chemically gated cation channels. All right, so these are shown in the picture here. Now, remember, I just want to mention the different types of channels because, once again, review this all the time. These are protein pores in the membrane that allow ions to move down their concentration gradients. Leak channels are always open, and we just talked about that for continuous diffusion. Then we have chemically gated channels. This is what we're talking about now, and this is what's found in the receptive segment. They are normally closed, but they open when a neurotransmitter binds. We can also call them ligand gated channels. A ligand is like a small chemical that binds. And then finally, we have voltage gated channels. We will talk about these in the initial segment as well as the conductive segment. And so voltage gated channels are normally closed, but they open when the membrane charge changes. And then voltage gated sodium channels have some additional special things we'll talk about next lecture. All right, so the receptive segment is part of a chemical synapse, right? So it's the postsynaptic cell, and it's usually receiving information from another neuron. That neuron sends the information by way of neurotransmitters, chemicals released from the presynaptic neuron. And so when a neurotransmitter binds to a receptor on the receptive segment, it creates what we call a graded potential. And as I said, we're going to talk about what a graded potential is as we go. So as I said, there are 4 different segments in the neuron, and the first one we're talking about now is the receptive segment, right? So this is where the binding of the neurotransmitter from the presynaptic neuron, the one here that's sending the signal, is going to, the neurotransmitter is going to cause graded potentials in the receptive segment. Then we'll have the initial segment, the conductive segment, and the transmissive segment. So we'll go over these segments one at a time. So the receptive segment is the first part. Graded potentials are going to be something that is found in the receptive segment. They are small, short-lived changes in the resting membrane potential. with the following characteristics. They are established in the receptive segment by the opening of chemically gated ion channels. We'll see how in a second. They're local, which means they only they don't move a long way, so they don't change the current in the whole neuron, just like in the area right next to those, or right fairly close to those receptors. And they're short-lived; they don't last that long, and they vary in the degree of change and the direction of change from the resting membrane potential. They could be small or large. They can cause what we call a depolarization, which means the cell could get more positive, or they can cause a hyperpolarization, which means the cell can get more negative, even more negative than resting. Okay, I'm going to switch to the whiteboard for a second, and we're going to go over a little bit about what happens in this receptive segment. All right, so for reference, this is going to be the other neuron. This is the actually conductive segment of the other neuron, the first neuron, the presynaptic neuron. Then we have, this is going to be the postsynaptic neuron. This is the receptive segment down here. And so the receptive segment is going to be either the cell body, which is also known as the soma, or it's going to be a dendrite. And so let's look at some of these things here. So we have got these are gated channels. This is a chemically gated potassium channel. Then we have a chemically gated sodium channel or cation channel. The cation channels are mostly sodium channels, so you can kind of think of it that way. And then we have a chemically gated chloride channel over here. And remember that gated channels always start out closed, and they only open for a moment and then they close right back up. All right, so what happens here, and this should be all a review from the neuromuscular junction. So we have an action potential that comes down the presynaptic neuron. The action potential is then going to activate this nice voltage-gated calcium channel, right? So put a little, lightning bolt over here. What happens to the voltage-gated calcium channel when the voltage changes? Well, it's going to open, right? And it's going to open, and calcium is going to rush. It's a channel, so it's going to passively rush down its concentration gradient. So remember, at this point, there is more calcium outside the cell, right? So we have more calcium outside the cell, and so it's going to rush down its concentration gradient into the cell. And so that's this is going to be the calcium. So the calcium comes into the cell, and it actually binds to the synaptic vesicles on the inside of the cell. And this binding of the calcium to the synaptic vesicles is going to trigger the release of these vesicles in a process called exocytosis. All right, and so exocytosis is going to happen, and let's do this one at a time. All right, so let's look at this chloride one first. So exocytosis is going to happen. Our synaptic vesicle is then going to bind. It's going to move over here, and it's going to bind with the cell membrane. It's going to merge with the cell membrane, and it's going to release the neurotransmitter inside. And that's this green neurotransmitter over here. And it's going to diffuse across the cleft, and then it's going to bind to this chemically gated chloride channel. And then the chemically gated chloride channel is going to open. So does anyone remember where there's more chloride? Is there more chloride inside the cell or outside the cell? Right, there's more chloride outside the cell, and so if this is a gated channel, so chloride is going to move into the cell. So we have chloride moving into the cell. All right, is chloride positive or negative? Chloride is negative, so we're gaining more negative charge inside the cell, so the cell gets more negative. And so when the cell gets more negative, we call it an inhibitory postsynaptic potential, right? Inhibitory PSP, inhibitory postsynaptic potential. And these inhibitory postsynaptic potentials are going to take the cell further away from threshold. Further away from threshold, because it's going to be less likely that the cell is going to be able to have an action potential because it's getting more negative, right? And we'll talk a little bit more about what threshold is too. Threshold is going to be, the cell has to get more positive. It has to get close enough, more positive in order to activate the opening of those voltage-gated channels. Remember, we talked about this also in our muscle tissue. All right, so when chloride comes in, it actually makes the cell more negative, which gets it further from threshold. So that is going to make an action potential less likely. We say it inhibits the action potential, and so it's called an inhibitory postsynaptic potential. All right, so next let's talk about this potassium. What's going to happen to the potassium? We'll do this one pretty quick. So, right, once again, we have exocytosis happening. We're going to have the neurotransmitter is going to get released. This blue guy over here, it's going to bind to our chemically gated potassium channel. And then when that happens, this chemically gated channel will open and it will allow potassium to move down its concentration gradient. So where is there more potassium, inside or outside the cell? Remember, potassium is like a puppy, so it's inside the cell. So potassium is actually going to be moving out. And I know we said before that potassium is a little bit conflicted, but still it's going to be, in general, moving out, down its concentration gradient. And so we're going to have a lot more potassium, moving out of the cell. All right, so is potassium positive or negative? Positive. Potassium is positive, so we're losing a positive. And so the inside of the cell is becoming more negative, right? And it becomes more negative. We call it an IPSP, right? It's once again, it's getting further away from threshold. An action potential is becoming less likely, it's being inhibited, so we call it an IPSP. And so finally, we have our last type of cation channel, which is going to be a sodium cation channel, all right? And so let's see, the calcium goes and it's going to bind to these synaptic vesicles. They're going to undergo exocytosis, and some of this red neurotransmitter is going to come out and it's going to bind to our sodium voltage gated channel. And that binding of the our sodium chemically gated channel, excuse me. And this is actually called a cation channel. This cation channel is the same as the acetylcholine receptor that we talked about in the neuromuscular junction. So this is the one that you're probably really familiar with, and maybe we should have started with this one. But so we have now got, when our neurotransmitter, which may be acetylcholine, right, when it binds to this chemically gated sodium channel here, or cation channel, a little bit, so it's going to let both sodium and potassium out, or let both sodium and potassium through, but mostly sodium is going to be moving. So if you remember, where is sodium? Where's there more sodium? There's more sodium outside of the cell, right? It's like snakes are outside, and so the sodium is going to want to come in, right? And is sodium positive or negative? Sodium is positive, and so this means that the inside of the cell actually gets more positive, right? That's what normally happens at the motor end plate, right? And so this is going to be excitatory. It's going to get our cell closer to threshold, and we're going to call it an excitatory postsynaptic potential, or an EPSP. And this EPSP is going to make the cell more likely to have an action potential. All right, so let's go back to our lecture. All right, so as I said, we have graded potentials in a postsynaptic neuron. We call them postsynaptic potentials. So that's exactly what we were just talking about. So these things right here, where I said it gets more negative or more positive, these are graded potentials that are being created in the postsynaptic cell. And so these graded potentials in the postsynaptic neuron are called postsynaptic potentials. Postsynaptic potentials that result in a depolarization, which means the cell gets more positive, are called excitatory postsynaptic potentials, or EPSPs. Postsynaptic potentials resulting in a hyperpolarization, which means the cell gets more negative, are called inhibitory postsynaptic potentials. And the numerous postsynaptic potentials can be generated because postsynaptic neurons can bind many neurotransmitter molecules simultaneously, right? So we have a whole bunch of inputs coming into these neurons. Some are EPSPs, some are IPSPs. So as a review, excitatory postsynaptic potentials are depolarizations caused by sodium entry, right? So those cation channels are going to open and sodium enters the cell. And this means that our cell becomes a little bit more positive. Inhibitory postsynaptic potentials, or IPSPs, are hyperpolarizations caused by potassium exit or chloride entry, right? And so that's either going to be opening of those chemically gated potassium channels or opening of those chemically gated chloride channels. Either of those cause the cell to become more negative. And this means that it causes this hyperpolarization, where the cell gets more negative than resting. And then we call that an IPSP because it gets further away from threshold and an action potential is inhibited. It's less likely to happen. And so we can have a whole bunch of neurons, right? A whole bunch of presynaptic neurons synapsing with one postsynaptic neuron, right? So we can see like 10 different neurons here, right? And so they're getting all of these are EPSPs, the green ones, and all the red ones are IPSPs. And so how does this neuron know what to do, right? Some are telling it fire, some are telling it stop. And so this is where the initial segment comes in. All right, so the initial segment is the next segment, and this is at the axon hillock. So all of these graded potentials from the dendrites and the cell body kind of come together and they all move towards the axon hillock. And in the axon hillock, something called summation happens. So, and like you can think of summation as adding, right? So math is like doing sums. We're summing things together. We're adding things together. So we are adding all the EPSPs and IPSPs together at the axon hillock to see if we can reach threshold or not. So we're adding the voltage changes from the dendrites and the soma together. So some might be positive, some might be negative, but we're going to see at the end if we get to that threshold number. Remember, in a muscle cell, threshold was negative 65 millivolts, but in a nerve cell, it's a little bit more positive. So threshold in a nerve cell is typically about negative 55 millivolts. And so the threshold is the minimum voltage change required. So if it changes to negative 40, negative 30, negative 20, 0, positive 20, any of those would reach, so that it reaches threshold or becomes more positive. So generally, you need to have multiple EPSPs added together to reach threshold. And the IPSPs kind of go in the opposite direction. They fight against those. And so once threshold is reached, an action potential can happen. So if threshold is reached at the axon hillock, which is the initial segment, then that's going to cause voltage-gated channels to open and the action potential will be generated. And we'll talk more about exactly the special types of voltage-gated channels next time. And you guys remember it from muscle contraction, hopefully also. All right, so summation can occur both across space and time. And so as we can see here, we have a lot of different cells that are going to be touching these neurons in different spaces, right? And so spatial summation is when multiple locations on the cell's receptive regions receive neurotransmitters simultaneously and generate postsynaptic potentials. And so we can see this is spatial summation. So we have all of these different areas. Let's say at exactly 1 o'clock, each of these different areas are going to, an EPSP or an IPSP is going to happen simultaneously. And so then, you know, if they're both EPSPs, you get a bigger EPSP. If they're both IPSPs, you get a bigger IPSP, you know, and they might cancel each other out if you have one EPSP and one IPSP. And so this is when we have multiple inputs at different areas of the cell. Then the next type is temporal summation, in which temporal, think of time, and this is going to be one single presynaptic neuron is going to be sending, repeatedly releasing neurotransmitters and producing multiple EPSPs within a very short period of time. And so this is a little bit of that example here, although it's hard to show. It's only one of these, like say it's just, you know, number 1 or just number 7, and that 1 is sending multiple little short bursts. And in reality, both of these are happening together. So you'll usually have both spatial and temporal summation happening kind of together. And this is another, this is not from your book, but this is another, I thought this is actually maybe a little bit easier to understand. So temporal summation, we have one neuron sending a bunch of little, you know, firing a bunch of times in a short period of time. Whereas in spatial summation, we have each of these, like let's say in this case 3 neurons, and they're each sending a pulse, and this is all, these 3 are occurring simultaneously at the same time. Then these 3 are occurring simultaneously. These 3 are occurring simultaneously. So these 3 from in different spaces are simultaneously sending kind of stimulating that neuron. And so in reality, we have a whole bunch of inputs happening, and you probably pretty much always have spatial and temporal summation happening together. And so the initial segment, or the axon hillock, is where all that summation happens. And so we're adding together all of these different EPSPs and IPSPs. And so if threshold is reached, the action potential is generated and propagated down the axon. We'll talk about that in the next video in the conductive segments. If threshold is not reached, we call this a subthreshold stimulus. The voltage-gated channels stay closed and nothing happens, right? And the graded potentials eventually, they're short-lived, so they eventually sort of disperse and it goes back to rest. The axon is going to, the action potential will be the same no matter like how positive the cell gets. So if the cell gets really positive, it'll still just trigger an action potential. If it just hits threshold, it'll still trigger an action potential. So all the action potentials are the same. And so kind of like your book likens it to what happens with a gun. If you try to pull the trigger but you don't pull hard enough, nothing happens. If you pull the trigger enough, to actually like fire the gun is going to fire. If you pull the trigger really, really, really hard and squeeze it really hard, the gun is not going to fire any faster, right? It's still just going to fire the same. All right, and then this is, I thought this is a nice sort of graphical example of what happens with summation, where we're adding the multiple EPSPs, then we subtract an IPSP, we add an EPSP, subtract an IPSP, finally we add another big EPSP, and then finally we get threshold. So once we get up to threshold, and this is, I guess ignore the numbers here, and then an action potential is going to happen, and then it eventually come back down. So that is all we're going over in this recording, and we will finish up on the action potential starting with the conductive segment next lecture.